Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. Today we are going to learn the hybridoma or the cell hybrid. In this we will take the two types of the cell. One is the splenocyte or all the cells present in the spleen is called the splenocyte. These are the cells that contain the plasma B cells, means the activated B cell. Those are going to produce the antibodies. So these plasma B cells are prepared by immunizing a mouse, and these are grown in the HPRT positive environment. So these are the cells from the spleen of a mouse that is already immunized against the antigen that we want the antibodies. Okay, and these cells are grown in the HPRT positive environment. There is another set of cells, they are called the myeloma cells, which are the immortal, and it is grown in a medium that is HPRT negative. So after that, these cells, the splenocytes and the myeloma cells, they were fused in the presence of the polyethylene glycol. So there is a cell and the cell fusion. So two cells become one cell, or the nucleus of the two cells will be combined along with the cytoplasm. So because of this fusion, there are the five scenarios that is possible. What are the five scenarios? There are the unfused plasma cells, means there is only one plasma cell. There is the fused plasma cell, so that we can see the two plasma cells together. There is the unfused myeloma cells, and there is the fused myeloma cells, means there are the two myeloma cells. They are fused together. And there is one scenario where actually the cells we need, that is it contains one plasma cell and one myeloma cells. So when I am saying plasma cell, myeloma cell, myeloma cell, plasma cell, myeloma cell, it means the nucleus. So this is a one cell with one nucleus. This is another cell with the B nucleus, suppose, and this is the A nucleus. So after the fusion, what it will become, the cytoplasm will fuse and the nucleus will fuse, so that this is just a bigger cell with nucleus A and the nucleus B. They are together, and these two nucleus will also fuse. So the situation means in the presence of the polyethylene glycol, the cells they get fused, means their plasma membrane they fuse with each other and it becomes one cell. So suppose there is the n number of the x amount is the amount of the DNA present in cell this one, and this is the another x amount of cell present in this one. So in the fused cells we have the 2x amount of the genetic material or the DNA. So these are the five types of the scenario. So what we will do next, we will grow these few cells in a HAT medium, that is the hypoxanthine, aminopterin, and the thymidine. So what is the role of this HAT medium? It contains the aminopterin. So aminopterin blocks the synthesis of DNA by inhibiting the dihydrofolate reductase. So it is a inhibitor. What it is? The dihydrofolate reductase, this is needed for the de novo nucleic acid synthesis. So when the HAT medium is there, so the cell cannot use the de novo nucleic acid or the DNA like the immortal cell. So this is a cell, this is means preparing its DNA by the de novo pathway. It is HPRT negative. So what is then HPRT positive? So HPRT is the hypoxanthine guanine phosphoribosyl transferase. When there is and it contains a thymidine kinase. So in the presence of the hypoxanthine and the thymidine, it can utilize another pathway, that is the salvage pathway, to prepare its nucleic acid. So when it is HPRT positive and it is grown in the HAT medium. So if the cell has this supplemented with the hypoxanthine and the thymidine, it can prepare the nucleic acid. So where the plasma cells are there, it is able to use the salvage pathway. And myeloma cell will give you the character of the immortalization. So in the presence of the HAT medium, these cells will survive For the unfused plasma cells, it can only use the salvage pathway, but it is not immortal. So in successive generations, they will die. The similar situation will be the fused plasma cells. So in several phases, these cells will die. Well, what is the unfused myeloma cells? It is immortal, but it cannot use the salvage pathway. So it cannot prepare the nucleic acid by the salvage pathway because we block the de novo DNA synthesis in the presence of the aminopterin. So in the presence of the HAT medium, these myeloma cells will die as well as the fused myeloma cells will die. So HAT medium is a type of the selection process in which we are actually selecting the cells which is a hybrid cells of a plasma cells and the myeloma cells because it will get the immortalized character from the myeloma cells and it will be using the salvage pathway to prepare the nucleic acid by the HPRT positive plasma cells. So in this way we were selected the hybridoma cell. So next what we will do, because we know that this selection process will take 10 to 14 weeks. Okay. So aminopterin is diluted from the culture by the several passage of the cell in the HT, the supplemented to the hypoxanthine and the thymidine one. It will take almost 2 to 3 weeks and before transfer into the normal hybridoma growth medium for the screening of the plasma cells so that we can get the identical antibodies. So now we have the hybridoma, we have suppose means A plasma cells, B plasma cells, C plasma cell. So this will produce the identical antibody, this will produce the identical antibody, this will produce the identical antibodies. But now they are present together. So it is a polyclonal antibody. So what we do in the screening, we make the single cell cloning of these all plasma B cell so that whatever clones will be selected, we will get the clones with identical antibodies. Means we can only prepare or amplify only one type of the B cell, either A, either B, or the C. And we will take the most efficient antibody that is having the high affinity to the antigen, and it is all identical. This is only possible because of the screening. So, hybridoma technology is a method by which we prepare the identical antibodies by selecting and screening the B cells, which is very highly having affinity toward a specific antigen. Thank you.